Yeah, Dr. Lisa, can I start now?
Yes, Prof. You missed that now.
You start the broadcasting now in Facebook? Okay. Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. It's giving us great pleasure and honor to welcome all of you today in our ICA webinar series number 9, Bioeconomy, Food Security, and Health Life. My name is Professor Hisham, Director of ICA Bago, and you are welcome to be with us today. Today is a very special webinar for us because I don't like to have a guest, but we have a family member who is just in Indonesia next door to us. And it's great pleasure and honor to welcome today Associate Professor Dr. Dalia Sukmawati. Dr. Sukmawati is Associate Professor and currently the Vice Dean of Faculty of Mathematics and Natural Science at Universitas Negeri Jakarta, Indonesia. She holds a doctoral degree in microbiology from the University of Indonesia, one of the top universities in the world, known for their high quality research in microbiology. She was appointed as the Vice Dean and focusing on international collaboration. Her research focus is in metabolomic of fungi, microbial fermentation, and microbial ecology. She has published over 76 journals, articles, and engaged herself in active collaboration. She was also a keynote speaker in many international conferences in Turkey, in Malaysia, in many different countries. She worked and contributed to interdisciplinary research and academic exchange, and is widely respected scientist in contribution in the microbial biotechnology. And I was honored to publish, I think, with her more than 50 index publications so far, Dr. Dalia. It was great pleasure and honor for this journey to have this more collaboration. Having a nice webinar need also to have an excellent moderator. We have today with us, he is technologist Dr. Zalina Binti Uthman, Noor Zalina Binti Uthman, and Dr. Noor Zalina, she received her PhD in bioprocess engineering and Master of Engineering from University Technology Malaysia. Dr. Noor Zalina is research interest generally involved in the application of bioprocessing strategies to improve productivity by combining optimization of production media, process variables towards scaling up of the food and agriculture industry. Dr. Zalina Uthman, she published, I think, more than 50, 70 publications so far. She was also speakers in IKI in the United States for a couple of times. And she is also invited speakers in many conferences. And her contribution in different fields of microbiology, industrial biotechnology, in wild stream microbes, recombinant microbes, enzyme technology is very known in the field. Therefore, I'm very happy to have today an excellent speakers and excellent moderators to have excellent webinar, inshallah. Thank you very much. Please, Dr. Dalia, the floor is yours.
Thank you, Prof. Okay.
Can you please share your slides?
Okay.
Open.
Okay. Alhamdulillah. Maybe Dr. Zalina, open your mic. We cannot hear.
Okay, sorry, Doctor. Okay, before we proceed, I would like to say thank you for Professor Hisham for his kind and very warm welcome for us. So, for not further ado, I would like to invite Associate Professor Dr. Dalia Sukmawati. The floor is yours now.
Thank you very much. I would like to share my presentation first before I speak. Okay, first. It is work, my presentation?
Yes. Yes,
Okay.
Prof.
Ya, Alhamdulillah. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning and warm greeting from everyone from Indonesia. It's truly an honor and pleasure for me to be here today at the ICA Webinar Series 9, hosted by the Innovation Center of Agri-Technology and advised by Processing ICA Universiti Teknologi Malaysia. I would like express my sincerely gratitude to Prof. Hisham as a director of ICA and my brothers, of course, since 2018, yeah, Prof. We already meet each other and so many publications, so many activity we all share with Dan, and thank you very much to sharing with me to give me invitation to share about the EcoFerment with yeast in innovative agricultural industrial waste management for alternative protein. For this concern that we know about today, I will sharing how microbial biotechnology, particularly yeast-based fermentation, can play a transform role in converting agricultural residue into high-volume protein-rich biomass, contributing a circular bioeconomy. I hope this season will inspiring and dialogue and collaboration. And I look forward to everyone who saw with our webinar to forward to exchanging knowledge and idea with all of you. Terima kasih. Let me start it for this kind of our discussion. As we know about our planet is under pressure, with very under pressure, with many pollutant, with climate change, for waste accumulation. So this is our challenging to protect our environmental. One is the problem is agro-industrial waste in particular contribute significant to this problem for the pollutant, for the climate change. The agro-industrial waste is very contribute. So we have to know what will we do, what will we have to transform it into available resources. One is the way is we use microbial fermentation to this circular solution. What kind of the agro-industrial waste? For the agro-industrial waste, we can find so many. For example, corn cob, molasses, sugarcane, soybean meal, corn steep liquor. From the industrial, we can find the waste of hulls, the straw, the grain, the meals. From the dairy industry, we can find the buttermilk, spoiled milk, whey, so many others. And for the fruit and vegetable industry, we can find rotten fruit, vegetable, peel, seeds, wastewater. It's very pollutant if we cannot use this one. We know about this all agro-industrial waste we can use because why? Because we know this is not just a waste, but they contain many substantial of product. For example, mineral, protein, asam amino. So what we have to do about this one? For example, industrial waste from sugarcane bagasse or SCB. SCB is very huge of bioactive compound. For example, they composing about the cellulose, hemicellulose, lignin, and protein. But before we use that for another valuable product, we have to do like a pre-treatment. What kind of pre-treatment we can done? For example, physical treatment. Physical treatment, we can grilling or we can milling, or chemical treatment, we can use acid or diluted acid or alkali treatment to break down the lignin or hemicellulose. That because this sugarcane bagasse is very polysaccharide, so we have to cut this one to a small piece with the pre-treatment. After we do the pre-treatment, we can use the biological treatment. We can do this one with the microorganism, for example, yeast. Why we use the microorganism? That's because the microorganism is very have beneficial. For example, they didn't produce inhibitory substance for enzyme production, and then they very fast for growing. For 40 hours, we can get the biomass of this microbe. And this is very minimal input energy requiring, and very good of this microbe. They can produce many enzyme who help to break down this polysaccharide substance. For example, if we do fermentation with the microbe, even though this is fungi or bacteria, they can produce many kind valuable nutrition enzyme. For example, lipase, lactase, cellulase, silanase. This is very great for industrial. So the waste from agro-industrial will not be a pollutant anymore, but we can convert very useful substrate. What kind of industrial waste? We can use oil palm empty fruit bunds. Why? This is one of our research in my research team. Because the oil palm is very huge in our country. As you know, Malaysia almost planting in oil palm fruit bunds too in our countries. So almost half of our countries is contained of the farmer of the oil palms. One is the waste of the agri-industrial. This is oil palm empty fruit bunds. My research is concerned how to make this substrate as a substrate for fermentation of microbe, and then feeding substrate for one of the insect. This is the black soldier fly. Why black soldier fly? I will tell you later after this presentation. The palm oil is very huge for the cellulose, hemicellulose, and cellulose is almost 45%. So this is a source of the carbon. That's why I concern to use the oil palm empty fruit bunds for the substrate of my yeast fermentation. And the others is pothacks. I don't know in ICA have not about this all of the kind of agro-industrial waste, but I hope it can be inspiring, which the industrial waste we want to do, we want to convert to make a good environmental heart. As you know, I'm produce just chocolate. I don't know, Prof. Hisham would still remember or not, or want to taste or not this chocolate. But this is a good chocolate, I think. I produced this chocolate from the bee. until the product, and it commercial Tokopedia. This is the online in our countries, just chocolate, and the problem is after I done the chocolate product, what should I do with the fruit buns? What should I do with cocoa pod husks? It is very waste. So the cocoa pod husks, it can be converted to into substrate to the insect feed or compost to reducing waste in chocolate industry. I hope will be one insight. So we not just produce the chocolate, but we can produce the waste of the pot husks to be another products that very useful. Okay, okay, and then why I use all of this kind of the waste industry? That's because we are from FMIPA UNJ, one of the largest university in Indonesia, Alhamdulillah, and our professor, Prof. Hisham, be a young professor in our university, especially in our faculty. Thank you very much, Prof. Hisham, for this one. And then I hope, as you mentioned today, we will do a lot of collaboration, many collaboration. And I want to tell you about, we have Universitas Negeri Jakarta Culture Collections. It's almost 6 years ago, 2014, I built this Universitas Negeri Jakarta Culture Collection, and it is member of UNMICRO and WDCM, one of the largest culture collection in the world that's in China, in Beijing. And we are a member that each year we invited to present to their forum what kind publication, what can we do about our microbe. We almost have 300 one isolate of microbe is contained of fungi, yeast, or bacteria. We concern about the focus biodiversity, health food, and agro-food industry. That's why ICA is very convenience and very strong collaborated. Thank you very much for your trust as profession and ICA member to be our partner. In our microbe, we already done, they have very potential, for example, inhibition, again, antagonist or biocontrol for fusarium, for many Phytophthora with very disease in our plant of the plantation. So this can be a biocontrol agent of this yeast or bacteria and filamentous fungi. And then for this resort, why I use yeast? Because yeast is my particularly major, and I'm very glad of the yeast. Yeast is very tiny microorganism with same with bacteria. We cannot see without microorganism. The cell is more bigger than the bacteria. It's almost 1.5 micrometer for the width and 5.3 micrometer in the length. The metabolism is very fast, and then we can harvest the yield of this biomass is only 24 or 48 hours of the yeast. And our favorite or our good potential is Pichia kudrazevi. Pichia masurica is it is a kind of the candidate of our Universitas Negeri Jakarta Culture Collection. The Pichia kudrazevi is a good producer for enzyme that produce pectinase, and for Pichia masurica, it's particularly good in alcohol and metabolite production. I have done these two kind of the yeast for enhanced volatile compound of cacao fermentation. I do two kind of fermentation of this kind of yeast. The cacao aroma for cacao fermentation is very good, but for today I didn't talk about the chocolate. I talk about the black soldier fly. This is the kind of value of the yeast. Even though first they produce of enzyme, they can be a single cell protein or SCP. Because why? Because they very contain of good asam amino. For example, alanine aminotransferase or aspartate aminotransferase (IST) or ALT. This is very good for improved liver function for the fish. So we can use this kind of the yeast for probiotic or for making good health for many others animal, for example, fish in the industrial farmer. So the others, the value of the yeast, they can produce many enzymes as I mentioned before. So why I concerning about the result of the yeast? And this is before I talking about my research, I know this is maybe for the audience. Thank you very much to join with us. I would like to a little explanation about before we use this yeast. You have to know there is two type of fermentation. For I want to explain. First is submerged fermentation, and the second is the solid state fermentation, or SSF. The submerged fermentations, it will be maybe a quite high equipment we have to use. We have to have the fermenter, a huge or a large fermenter, because in the submerged fermentation, it contain of the water or liquid medium for the submerged fermentation. But the beneficial of the submerged fermentation, it will be very fast and very controlled. And the second is the solid state fermentation. The solid state fermentation is cultivation using microbe for with the absent or near absent for flowing water. So it very convenience us to do the solid state fermentation to convert the agro-waste industrial to other valuable industry. This is for example substrate used in solid state fermentation, like we can use sugarcane bagasse, tea waste, wet bran, or cocoa pod, or from the oil palm branch. So this is the substrate. And then this is the floor of the fermentation. For example, we have the microbe, even though this is yeast or the fungus or the bacteria, and then we do inoculation preparation with a lot small, and then we have to decide what kind of substrate we want to. It is the crops or extract concentrate or agri-food industrial bioproduct concentrate. And after that, we can do the fermentation. We put the microbe there with the condition. We have to know what is the optimals, what is the pH, what is the temperature, what is the great inoculum. There's so many parameters we have to do if we want to get the good value or the good product of this kind of fermentation. For example, the fermentation, after the fermentation done, they will convert to organic acid, biofuel, antibiotic, aromatic, biopesticide, and so many others products. And one of my concerns I do about the Black Soldier Fly Larvae or BSL, or in the Latin is Hermentia illucens. The Hermentia illucens is wait. The Hermentia illucens is a black soldier fly. We call it black soldier fly. This is very huge insects, very fast with 45 scale. And then I already learned about the black soldier fly. It's very strange opportunity because why? Because this larva of this kind of the insect is contain almost 40 or 60% of the protein. So it will be converted to to be a protein, to be alternative protein for the next generation. So I already done about the oil from the larva of the black soldier fly. They have antimicrobial activity. It's still in progress. This is the picture of the antimicrobial I already done, and it's very good. So this is very challenging using black soldier fly, using many kind of the yeast, many kind of the microbe to know about what kind of product of this one. I would like to give you brief or insight. One is this is the adult of the insect, and for the male, they will die after mating, and for the female, they will die after laying egg. They will die. So this is they have very dedication. This is black soldier fly is very dedication organism. After they do their job, they will die, even though female or the female. This is the egg of the black soldier fly. They can produce one adult of the insect of the black soldier fly. They can produce 400 or 800 eggs. And after this egg, I will put to the some medium for our substrate to make egg to the neonate larva. It's almost maybe in 10 days. And after the 10 days, there will be a feeding larva like this one. This is the feeding larva. It's very small, it's very cute, and it's very great for I don't know, but I'm not yet eat that. But this is good for fish, for the many other and many other farm, maybe other animal is very good as a source of protein. After feeding larva, maybe it's almost 20 days, there will be a pre-pupa. It's need 10 days more, and then after pre-pupa, there will be a pupa. The pupa is very dark with the very great chitin. The skin of this larva is very great of the chitin. Chitin is very huge source for the antimicrobial substance. I have done for this one, and Alhamdulillah, I have one of the student. They already published with some of paper about the chitin from the pupa, and after the pupa, they will be adult, and this is the whole cyclus of black soldier fly, where we can use for the agro-industrial waste. What states? In the feeding larva states. In the feeding larva states, we can put the microbe there with using agro-industrial waste. For example, like this one. I would like to give you a brief about how we done about the preparation grow of black soldier fly. This is one of my student. They do for the substrate selection. They put in the cut a small piece because the larvae didn't have tea. They just absorb the kind of the waste of product. And after that, we put the substance application with suspension. This is like a suspension that we have done. And then after this, we do the incubation. Why we do inoculation of the yeast? That's we concerned, but the yeast can break down and can be at the valuable of the nutrition of this for the black soldier fly. The yeast have some amino. The yeast have good biomass. The yeast have good enzyme. So we thinking about the valuable of this nutrition after 3 days fermentation will be a good tool for feed of the black soldier fly. And this is kind of the basket for the larva of the black soldier fly. 1 gram or 10 gram of the substrate can be 1 gram of the larva can convert 100 gram of substrate. Can you imagine if we have a ton of the waste product, they very fast they can do the black soldier larva is very great army for making this kind of ways to be a good value. Like this one, I give you this is the larva with the small. They convert the kind of these substrates to small and very very tiny. And then after this one, we can harvest even though the frass or the larva itself. So it will make me maybe it's almost 20 days fermentation. We can harvest of this one. Okay, I would like to share. This is one of our students with Prof. Hisham, or this is already done. Thank you very much, Prof, for your kind of the suggestion for very good in our research. I do Pichia cecembensis and Aspergillus nigers using pot hugs to enhancing black soldier fly larva. And then we have to done, this is the pot, and then we put the inoculation of the yeast and the Aspergillus niger, and then we see what is the result. This is the Pichia cechebensis and Aspergillus niger, and then we put this one, and then we know in 3 until 9 days fermentation, and we see what is the larva, what happened with the larva of black soldier fly. We know about the pH temperature, and we concern about the protein. And then we see for the parameter like of the larva, it's very good in even inside the control. And then we know about this one, the larva is very fast, more than fast, and the growth is more than very good inside the control. So about this one, and then we know about in the we see, and then we do the analysis proximate. We see the protein content fermentation and with the Pichia cecembensis and Aspergillus niger, and the protein is more better than the control, and it's almost 42 days. I think this is maybe for this one is not very quite different, but this is one of the solution for the waste product. This is not just to put in the garbage and make the pollutant, but we convert with a good with this one for the black soldier fly larva. And in the conclusion, with the fermentation, with the fermentation, the growth of the leg of the larva is very fast, and for the nutrients fermentation for the protein is very fast too than the control. This is our results, And we do for the Pichia cecembensis 2 and Pichia masurica for the growing chicken carcass quality. What is the happen? I want to know what is the happen for the boiler chicken boiler chicken if we put they with the maggot larva. The larva, we have to fermentation with the microbe, with the Pichia masurica and Pichia cecembensis. So the result we already done for the, and this is the under review too, and this is collaboration with ICA and our university, the impact of Pichia, and this is under review. Alhamdulillah, it will be submit maybe in next month. And what is the result? The result is before we done, we know about the antagonistic and we do a toxicitas. These two kind of the yeast doesn't toxic and good in the substrate with not synergic activity. And then what is the result? The result is very good. For example, in 40% Pichia masurica and Pichia cecembensis, they have good crude protein. In control, just only 38%, but with the microbe, the crude protein is very huge, 44.30% of the protein. The as content, the crude fibers is very the crude fibers and the crude fat, it's very good too for the alternative for the feed substitute of this kind of product. And this is the crude protein. The 40 masurica and cecembensis with the combined, this they can increase the protein content 44.30% more than the control. So this is what kind of alternative. So we can use the fermentation doing yeast with the high protein content contribute for good nutrition quality for the substrate. And then in our research that mentioned for the media boiler chicken carcass, almost 36% it will exchange, and for the weight of the chicken carcass it enhances too. So this is one of the alternative that we have already done a proof for the fermentation using waste product is very useful. And the others our research is we do Pichia kudrazevi for palm waste exchange for BSF grow for frass and potential fungi ecology. We do this is for the palm oil buns for the not using, but we use for substrate of the microbe, and then we do fermentation with Pichia kudrazevi. About this one, 42% Pichia suspension, they have a good for weight for the larva, and this is good improvement, positive effect for yeast fermentation and change substrate digestion and larva nutrient assimilation. So this is the BSF larva performance, more good than the control. And for the frass quality, after they have eat all of this microbe, they eat, and then we do the frass. The frass is we do for the antagonistic fusarium. Fusarium solani is one of the biggest problem in the banana trees in our countries. Using this frass for fermentation with Pichia kudrazevi can inhibit it, the growth of Fusarium solani is almost 35, almost 44% that can be done. And the composition frass can be a fertilizer. We know about the frass from the fermentation with Pichia quadrazevi. It contains nitrogen, phosphor, potassium, and many others. It's very good for good fertilizer. So this is, I hope this is the information for all of you, and it will be our save our planet with a good using waste product with fermentation. Even though I'm not just doing the fermentation, just done about the product of the protein, but I concern about what is the microbial diversity in the large of the BSF. So we do metagenomic analysis. The fungal community associated with black soldier fly, in larvae and frass, it's very quite different using Pichia quadrazevi condition and without quadrazevi conditions. This is the control. Yeah, in the left is control, in the right is the with the Pichia quadrazevi. That so many microbe with a good potential, with a good for health of the gut of the black soldier fly. This is the answer why black soldier fly with fermentation with good growing in the weight, growing in the large, growing in the body, and good substance of the protein. That's because the microorganism is very quite different too. The Pichia quadrazevi is influence with the diversity of the microbe. And this is the kind of the Pichia quadrazevi fermentation of oil plums, empty fruit buns is significant change the performance as nutrition quality of BSF, and the control we can comparing the control and the treat maggot. That's for example. Pseudophila marina is very good for the metabolism. They have good in enzyme too. So this is our changing to do the more result about the using fermentation yeast to others any product of the fermentation. And the conclusion is the study highlight potential of eco-fermentation using yeast is very innovative, very transforming industrial waste to very high value. So this is a change for us to do the small thing, to do the waste, for example, to save our planets, with do the something, the small step using the fermentation with yeast is very good process for agro-industrial system, represent a scalable and economical friendly solution. And this is my last of my presentation. This is funding of international collaborative community for services from my university, Universitas Negeri Jakarta. And thank you very much for my team, my students. This is the Roro, Hasana, Karina, Sinta Dewi, Lydia. This my great student. They have winner to the kind of Kitosan black soldier fly as fusari as antigenic antimicrobial for Fusarium oxyforum, and they winner to Indonesian students, and I'm very proud for this one. And this is my team, my co-team. Thank you very much. Thank you very much for this. I hope it will be useful for us.
Okay, thank you very much, Prof, for your insightful and inspiring sharing. And also want to say congratulations to your students for their effort to help you for your research activity. Now, but before that, I want to inform all of our audience, we have pre and post quiz. So kindly please go to our Facebook there is a Google form there, and you can answer the all of the question. Okay. Okay. Now we will proceed with Q&A sessions. Okay, Prof, you are ready for the questions?
Yeah.
So we have question 1 from Encik Muhammad Farid. What are some challenges in a large-scale black soldier fly farming, and how can they be addressed?
Okay. What is the challenging about the black soldier farmings? First is we have to know what kind of waste product we want to, because the black soldier fly, as I mentioned as I mentioned before, they can eat every kind of waste product. First is we have to know about what is the waste product we want to. But if we want to good product for the waste product for the good protein, so we have to find what is the waste product. It is content of the protein. If we want to make the black soldier fly is content of the chitin, so we have to use the black soldier waste product is from the chitin source. So we have to decide about this one. And the second is the challenging is for the pre-pupa, for the pupa, pre-pupa, and insect. This is the challenging too. After the adult of the insect, is this larva will be produce the egg or not? Sometimes the black soldier fly doesn't produce the egg. They just they mating, but they didn't produce the egg. If
Why?
they
Why,
didn't,
Prof? Why? Why?
why?
Tak
Yeah,
bahagia, tak
neither.
bahagia.
Tak tahu, mungkin nggak bahagia kali ya. So that's a challenging of how to this black soldier fry with mating with a good egg, and then with the fast egg. This is the challenges. What if the problem I using the peel of the banana and pineapple. So I do the result. We use kind of the banana, pineapple to be a good aroma, volatile compound. So this is the change, and then it will be work for the egg. That's the problem. If we doesn't do the completely cycle, so it will be our problem too. Because why? Because the black soldier fly is very short, just only 40 days. That's the problem.
Okay. Means that the substrate for their growth is also important.
Yes, of course. Yes,
Yes.
definitely, Lisa.
They don't like food waste as their substrate.
They like,
Our
but
kitchen.
but we have to know about what is the moisture, what is the content of the water, not too much. Because if we use waste product from the market, for example garbage, for example, or papaya, or carrot, this is very huge of the water, so it will be inhibited of the growth of this larva. So that's the kind of challenges to make this black soldier fly larva.
Normally, at what moisture content? The best
It's
moisture
all
content for this fly.
okay. It's almost they need just only 40 or 50%. All is enough, not
Oh,
much. If
okay.
much, it will be very wet, and this is not good for
Mm,
growing for the black soldier fly.
mm. Prof, you have tried to ferment the maggot from the black soldier fly with Saccharomyces cerevisiae instead of Pichia?
I already done Pichia kudrajevi, Pichia cicembensis, and Saccharomyces cerevisiae. All of these years I already done, but which one is the best? The best is if they have contain collaboration, synergism. not only just one, but maybe for
Okay.
example, I already show to you Pichia cecembensis with Pichia masurica. It is contain 40% of Pichia cecembensis, 60% of Pichia masurica. Like example like that. Not if just only one, the product of the protein is not very good.
Okay. But we still can use Saccharomyces cerevisiae itself. Yeah,
Sure.
yeah,
Yeah.
because uh, uh, for myself lah, I've never used Pichia. I don't know where to isolate it. Last time we just used recombinant Pichia pastoris, but this time I just wow, there is another Pichia.
Okay.
Okay. We have
Sure.
second question, also from Muhammad Farid. What are the
Mhm.
current regulation? Example, FDA or from EU on
Oh,
using black soldier fly larvae in animal feed or human food?
yeah. For black soldier fly, I think there is not yet, yeah, for human, because it's very rule regulation of FDA. Maybe yeah, we have to concern. So maybe for my research, and I read from many literature, the black soldier fly is be a good application for animal, for fish, for lamb, for not for human. That's
Last time I noticed there is one paper they try to make it as a protein for human. What
oh
is your
yeah.
opinion? For us, it's macro.
Yeah, I don't know because if you go to Thailand, not
uh,
even the soldier fly, they eat everything, isn't it?
yeah, they eat everything. Yeah,
Yeah, they eat.
as long it's not
Yeah,
poisoning.
yeah. But
Uh,
one is the concern I want to mention to you. Black soldier fly, they eat waste product, isn't it? Waste product.
Waste product.
Yes, waste product is contain many pathogenic microbe
Mm-hmm.
that contain many pathogenic microbe that produce mycotoxin. Mycotoxin is cancer stimulation in our body. So
Mm-hmm.
that's why I do fermentation first, that to decrease the pathogenic microbe to eat this black soldier fly. So if you eat a larva of black soldier fly that contain pathogenic microbe in his body, so it's very dangerous for our, isn't it? So why I do the fermentation first? Why? Because the yeast they are used, they have potential. For example, they can produce enzyme pectinase, they can
Mm-hmm.
produce cellulose, they can produce as a probiotic. So the growing of the pathogenic microbe for the gut of the black soldier fly, it will be reduced, and I hope there is no pathogen, even though for our human or for the animal. But for fermentation, waste product using microbe is not regularly in farmer, isn't it? Not very farmer doing the fermentation. They just
Yeah,
put any kind of waste and then they put black soldier fly. That's the dangerous. That's
yeah.
why I concern to not use this kind of the black soldier fly for the human.
Because from my opinion lah, because the farmer don't like to ferment it, the agro waste, because maybe they think after we ferment it, the aroma is more worse. Maybe
Okay,
they thought, yeah,
okay.
that
That's
is my opinion.
yes. That's I want to tell you about the aroma. As I told you, one is the biggest big and good for the yeast. They produce volatile compound. For example, ester, and then alcohol, ethanol. The smell is make the garbage is not very smell anymore. The smell is good if we do the fermentation. It's
Yes.
very quite different.
Mm, mm.
That's why we have to ferment it. But when the difficult for the farmer, they want very fast to take the money.
Yes,
Is it?
correct. Yeah,
Yeah.
yes.
So
I'm also
that's
uh,
our yeah.
yeah, I also tell my student, okay, the
Mhm.
fermentation is fail when you smell in our Malay we call longkang, a very bad smell. But if the
Yeah.
fermentation is good, you will have a very nice odor.
Yeah.
Betul
Yeah.
tak, Prof?
Betul. Betul.
Okay.
Betul.
So we have third question from Kasih Putri. Good
Yeah.
morning, Doctor. What do you see as the biggest barriers to implementing eco-fermentation technologies in rural or low-resource regions? This is the issue that we discussed just now.
Okay. Yeah. What is what
Uh,
is
what do you see as the biggest barriers to implementing eco-fermentation technologies in rural or low-resource regions?
Okay. The biggest barrier to implementing the eco is maybe for the our knowledge about considering to take our environmental. For example, climate exchange and very much garbage in everywhere. So
Mhm.
the barrier is from ourselves first, yeah. Is for example, maybe in our lecturer, I'm a lecturer, not in lecture is many just one publication, just only publication is okay, there is no product for the society. So this is the biggest problem. Many researcher in countries, maybe other countries, doesn't thinking about what is impact, what is his impact for the society. Maybe from ourselves, from my student maybe who watch our webinar, I hope we join together. We have
Yes.
what we have to do, what is our value for our environmental. That's the I think.
Mhm.
Yeah,
Mhm.
Dr.
Mhm.
Zalina.
Yeah. So, question from the audience? Okay, I have last question. Okay, during your you ferment with PKI, the PKI you buy the set or you isolate by your team, the PKI.
Okay. Thank you, Dr. Zalina. It's very nice answer. Question is very nice question. As I mentioned to you, we have Universitas Negeri Jakarta Culture Collection. We have
Mm,
almost 300 of the microbe. In our department, I'm from biology department, we concern about the biodiversity. So in my major, I always isolated from many kinds of indigenous food from Indonesia, or from indigenous fermented food from Indonesia, from fruit from Indonesia. So we done with ourselves collect isolation and then identification, and we collecting in our culture collection, and we have that kind of the yeast.
can we buy the strain?
No,
Ah,
bye. Just ask Prof.
free.
Hisham.
Free.
Prof. Hisham. ask, ask our director, okay?
Oh, bro, what? Another question. I promise this one is last.
Okay.
The fermentation of the maggot. How about
Mm.
the of the chitin? Because when we give this fermented maggot to chicken, apa, Black Soldier Fly maggot, the problem is chitin. The chicken
Mm.
cannot accept it if more than 30%, 20%.
Ah,
So ferment it, PKR, then the concentration will decrease or how is work?
okay. First, first is for the chitin. Chitin is very high instead of pupa and prepupa, doctor. So if
Mm.
you want to eat or to make it for the chicken, not
Uh-huh.
in that state, but in the feeding larva states, maybe is 20 after we do fermentation. The content of the chitin is very low, and it is
Ah,
not very huge and not very. It's good for the chicken. So
so
if
not a big issue
but
lah.
but if you give the chicken in the state of pupa or pre-pupa, it's very
Uh-huh.
huge of the chitin.
Okay. Doctor, we still have question in the chat box.
Yeah.
Okay. Okay, Dr. Dalia, based on your presentation, among the agro-industry waste you test, like palm anti punch, cocoa pod husk, tofu dregs, which show the highest improvement in protein yield after fermentation?
Show
It's from
show
Zulaika.
the highest improvement protein. Oh, okay,
Ah,
okay.
which
Yeah,
one is
thank you.
contain high protein?
Thank you, Zulaika, for a very nice question. What's the highest improvement in protein yield after fermentation?
Mm-mm.
Uh, okay. In this kind of it's very nice question. Yeah. Which one? That's in my best experience. I use cocoa pod. I use oil palms, and from the industrial waste, it's not just contain of this one, but for the best in my best experience is combined from the it's combined, not just waste this product, because the larva of black soldier fly like us, they need carbon source, they need protein, they need nitrogen, so we have to combine this one. And the key is which microbe we use. For example, if I use Pichia quadrazevi and Pichia masurica in the cocoa pod, it's very good for the yield, but it's not very good for the oil palm. Why? Because we have to pre-treatment first. So in this question, I cannot answer, but in our result in right now, the cocoa pod with combined with industrial and for the market, it's very good for the good yield.
Doctor, protein content is contributed by
Yes.
the yeast.
Sorry, I
The
think
the
this.
the protein content. Hello.
Yeah. The protein
The
content.
protein
What?
content is contributed by the yeast or by the.
Okay. The protein content is could be from the it could be from the many sources. Yeah, first, of course, from the yeast first, and the second, the protein content is from the waste product itself. I think they contain protein too. For example, lignocellulosic, for example, cocoa pod is have 30% of protein, so it will be combined for the crude, because I measure for the crude of the protein, not which one of the protein, but in the biggest highlight is uh, all of the fermentation, the protein is higher than control.
Okay, Doctor. Last question in the chat box. What recent technology advancement have improved black soldier fly farming? Example, is it automation, breeding optimization? And they also request you to visit ICA Pago in the future.
Of course. Thank you very
InsyaAllah.
much. Inshallah. Thank you very much. It will be glad for me to see ICA Pago, yeah, to see, because I already go to UTM, and for sure I want to go to
Last time you go
see.
to UTM Skudai.
Yeah, to Skudai, to
Ah,
IBD.
IBD.
Yeah. Okay, what recent technological advance have improved
Ah,
black soldier automation breeding automations? Maybe for this one, first is we almost done. We do the immobiles of this yeast. So we have immobilization using freezing, using gluten, using alginate, and using many others method to take this kind of the microbe immobiles of the yeast to implement to make very fast of automi for breeding of automation of black soldier fly. In our next generation study or NGS, we already see the pathogenic of the microbe, the pathogenic of the fungi. There is no if we implementation for the this kind of the yeast. So that's in the advance we will prove about the how can we do the immobile so we can share very much without suspension anymore. So we will do with the immobilization of the yeast and the enzyme of the microbe to improving the fermentation of black soldier fly. Maybe that's I as that I answer, Doctor.
Okay. For that, thank you very much, Prof, for
Yeah.
your knowledge sharing today.
Yes.
And thank you all for your participate with us. And now let's open your video for a quick picture together with our speaker. Or maybe our pengarah want to say something to our guest speaker today.
Yeah, first, I would like to say thank you very much, Dr. Zalina, for excellent moderation of the session and very critical questions as well, because I know you do also research in this field. And
course.
thanks for PM Dr. Dalia Sukmawati today for her time, for coming and sharing her knowledge openly, even though you disclose a lot of information today, which is behind the scene, not written in your literatures, I know. And we
Yeah.
want to share with all speaker, all audience here.
Yeah.
I see the question of Mr. Muhammad Farid for when asking you when you will come to see us in Bago. I would like to say for everyone, Alhamdulillah, we have now more than, when I checked, we have more than 80 joint publication with Dr. Dalia Sukmawati, index publications. And I think Dr. Dalia should to come to celebrate together 100 joint publication with UTM.